I think we'll go ahead and get started. So this problem that I've put up here, I'm going to see if I can get the zoom a little bit better on this one. So this was pulled from last year's AP exam, and there are a few things to make note of here. So in this one, researchers studying patterns of gene expression in mice. This actually deals with stuff that we had just been looking at, so transcription, translation, things that we had just been looking at before we went to distance learning. And they gave us kind of a strange figure here. So we've got 6 different genes along the top here, and we've got 6 different kinds of tissue that these are found in. And they've categorized it, either there is no mRNA expressed, a moderate amount of mRNA, or a high amount of mRNA expressed in these tissues. So looking at that, we're going to be using that as the information which we base everything off of. And they ask us 2 questions. First, we need to identify the gene that is most likely to encode a protein that is an essential component of glycolysis and provide reasoning to support our identification. The second part, the researcher observed that tissues with a high level of gene H mRNA did not always have gene H protein. Provide reasoning to explain how tissues with high gene H mRNA levels can have no gene H protein. So we're going to be breaking down those tasks kind of one at a time here and looking at what we can do there. So for that first task, we need to remember that glycolysis is going to be a part of metabolism. And in particular, that part of metabolism is going to be the splitting of sugar to make ATP and to provide pyruvate for the Krebs cycle. So we would expect then that all tissues should be making this protein. All tissues are going to need to perform glycolysis. All tissues are going to need to make ATP. So we would expect that all of the tissues are going to be making this protein. So to figure out which of these proteins is likely going to be essential for glycolysis, we're going to look here, and the only one that has expression of that gene in every kind of tissue is going to be gene G. So what we're going to say here is gene G is the only gene listed that is expressed in all tissue types. Therefore, it is the gene that is most likely to encode a protein that is an essential component of glycolysis. There we go. Typing, talking, and thinking at the same time apparently beyond me. So that kind of is the approach that we would take for part A. For part B, we are looking at why a tissue with a high level of gene H mRNA might not always have that gene H protein. So this one is a lot more open-ended, and for that part, we're going to be looking at this one. We can kind of speculate. So as long as we justify what we do with some solid scientific reasoning, we can make some speculation here. So the thing that we need to remember here is the way that we get from mRNA to protein, we need to have the mRNA leave the nucleus. It needs to bind to the ribosome. And the tRNA is going to extend the amino acid chain. After that, there might be some kind of post-transcriptional modifications that happen to the protein. So the places where this could break down, it might be that for whatever reason, that mRNA doesn't leave the nucleus. Either it gets broken down before it leaves, or it is blocked from leaving. Somehow it doesn't make it out of the nucleus. It might be that for whatever reason it gets out of the nucleus but fails to bind to the ribosome. It would be very difficult to break this process where tRNA extends the amino acid chain without breaking the rest of the cell, so that's going to be unlikely. And our last thing that would be possible is there's some kind of post-transcriptional modification that takes place. Some proteins, once they're transcribed, they get kind of melded together with another protein. Sometimes they are going to be trimmed or snipped in some way. So we've got a variety of things that might happen there. So for this question, it is a little bit open-ended, but according to the College Board, and this is a place where I actually do agree with what they say, kind of the sorts of answers that we might have are first, the mRNA does not leave the nucleus. Second, the gene H mRNA is not translated, or the protein encoded on gene H is modified after oh, actually, I apologize. I got this backward. So, excuse me just a second. Shows that I've had a long day here. Those post-transcriptional modifications, that would actually be over in this section here. So the mRNA transcript is modified before translation occurs. So I apologize for the confusion there. I read that problem a little bit too quickly and started saying things that were not exactly accurate there. So the three possible things that we could have, and you only need one of them, you don't need all three. One of them would be the mRNA doesn't leave the nucleus. The second would be that the mRNA does not get translated. And the third possibility would be that the mRNA transcript is modified before translation occurs. So what we've got here, this is going back to some biochemistry. So we're getting to properties of water. So one of those properties is that water, they say it is cohesive. And they're asking us to define and explain the chemical interactions and properties that make water cohesive. So first thing that we want to do is figure out what is actually meant by cohesion. So going way back to our biochemistry unit, cohesion would be the property where water sticks to itself. So cohesion, this is the property of water sticking to itself. In other words, putting it more scientifically, water molecules attract other water molecules. So why this happens, this is where we need to remember that water is a polar molecule, meaning that the electrons are shared unequally between the different atoms. So this gives it a partial charge to the hydrogen in the water molecule, and it gives a partial negative charge to the oxygen. Opposite charges attract. So the oxygen of one water molecule is going to be attracted to the hydrogen of another. Also, as part of that question, they had the characteristics of water are of central importance to the arrangement of the cell membrane. Describe and discuss how it is that water exerts this effect. So for this one, I'm going to be looking at basically why is the cell membrane the way that it is. So first, we're going to need to describe the structure of the cell membrane. So the cell membrane is composed of phospholipids. And there we go, fix the typo. These have a hydrophilic head, and that is the phosphate group. And a hydrophobic tail. That's going to be the lipid part of that. The phosphate, because it is charged, or because it is polar, I should say, is attracted to water molecules. The lipid, in which the electrons are shared equally, is going to be nonpolar. And it won't be attracted to water. So basically with this one, and I'm going to see if I can pull up an image of that. So because the heads are hydrophilic, they are going to be going towards the water, and that is going to put them on the outside of what's essentially a phospholipid sandwich here. So the phospholipid, like I said, it's got a hydrophilic head, a hydrophobic tail, the extracellular space and the cytoplasm being both primarily water. That's where the hydrophilic heads are going to go. In order to avoid the water, those hydrophobic tails are going to go in the middle, giving us that phospholipid bilayer. And really, what we need to explain and be aware of is that because the heads seek water they will face towards both the extracellular space and towards the cytoplasm, because the tails are repelled by water. They will form, they will be on the inside of the phospholipid bilayer. So it was a little bit of biochemistry for us. Now we've got a question about hemoglobin. So hemoglobin is responsible for oxygen transport in the bloodstream. Describe the levels of protein structure that make up a complex protein such as hemoglobin depicted above. So this is reminding us about, this is where we had primary, secondary, tertiary, and quaternary structure. And I heard and sensed the cringe from a great distance going back over this. So, primary structure, that is going to be the sequence of amino acids. Another way that you can put that is you could say it's the order of amino acids. That secondary structure is going to be composed or formed by hydrogen bonds between the amino groups and the carboxylic acids in the polypeptide. In other words, and now that I've got my technology working for me, going to pull up a nice image of this so that you don't have to watch me draw a thing. Let's see. There we go. All right, so the secondary structure there is going to be independent of the R groups. So notice that we've got these R groups for the protein coming off on each of those amino acids there. Those hydrogen bonds that we deal with are going to be between the amino group there and the carboxylic acid, that C double bond O, on another part of that strand. So they either make what's known as an alpha helix, where that structure kind of twists around, or they make what's known as a beta pleated sheet, which is going to be a structure where it kind of forms parallel bending lines that go through. So there's our secondary structure. The tertiary structure, that is going to be various interactions between R groups. So these would be things like hydrogen bonds between them. We might have hydrophobic interactions. Some of the amino acid R groups are in fact nonpolar and hydrophobic. We would have, this would be where disulfide bridges come into play. So if you've got something that has a sulfur group in it, there might be bonding between those. And let's see, so we got hydrogen bonds, hydrophobic interactions, disulfide bridges, and there might also be covalent bonds that form between there. And then the quaternary structure, that is going to be where multiple polypeptide chains come together or interact. And then for part C on this one, I'm actually going to skip part B. Part B is not all that helpful for our purposes. But for part C, this is more along the lines of the kind of thing that they'll probably be putting on the exam for you. So the good news and bad news with the exam format this year. Because they know you're taking it at home, they are going to be expecting you to use notes, use your textbook if you've got it, to use kind of any resources that are at your disposal, as long as you're not communicating about the test with anybody. So if you are pulling up your notes, looking at those things, all of this kind of recall level stuff should be in your notes or someplace handy where you can get to it pretty quickly. The kinds of things they're going to be asking you folks to do are explaining, describing, and making inferences or insights based on what you do there. So with this one, we're looking at explaining what is happening at the molecular level when hemoglobin is heated to an extreme temperature, and then describe the effect that this event will have on the ability of hemoglobin to carry oxygen. So first thing to be aware of, heating something up, it can break weak bonds. So this would be particularly hydrogen bonds and hydrophobic interactions are going to be particularly vulnerable to this. So once those bonds break, those bonds are responsible for keeping a lot of that structure together. So disrupting these bonds will change the secondary and tertiary structures there. And changing the structures changes the shape of the protein. And will change its properties or function. Because hemoglobin's function depends on its shape, heating it to extreme temperatures will reduce its ability to carry oxygen. Because the exam format is changing this year, I can't say exactly that, oh yeah, you're going to get a problem like this, but this is one of the meatier problems that they had on last year's exam, and they give you a lot of information. So one of the things that I recommend, especially on this exam where you're going to be taking it at home, is to go ahead and look at the entire question before you start trying to answer things. So let's take a look here. And this says, auxins are plant hormones that coordinate several aspects of root growth and development. Indole 3-acetic acid, IAA, is an auxin that is usually synthesized from the amino acid tryptophan. Figure 1. There it is. Gene TRPT encodes an enzyme that converts tryptophan to indole 3-pyruvic acid, I3PA, which is then converted to IAA by an enzyme encoded by the gene YUC. Circle one arrow that represents transcription on the template pathway. Identify the molecule to be absent if enzyme YUC is non-functional. Predict how the deletion of one base pair in the fourth codon of the coding region of gene TRPT would most likely affect the production of IAA. Justify your prediction. And explain one feedback mechanism by which a cell could prevent production of too much IAA without limiting I3PA production. Rhizobacteria are a group of bacteria that live in nodules on plant roots. Rhizobacteria can produce IAA and convert atmospheric nitrogen into forms that can be used by plants. Plants release carbon-containing molecules into the nodules. Based on this information, identify the most likely ecological relationship between plants and rhizobacteria. Describe one advantage to the bacteria producing IAA. And then part E, a researcher removed a plant nodule and identified several cheater rhizobacteria that do not produce IAA or fix nitrogen. Describe the evolutionary advantage of being a bacterial cheater and predict the change in the bacterial population that would cause the plant to reduce the amount of carbon-containing molecules provided to the nodule. Okay, there's a lot there, and these last two, because of the disruptions we've had to the school year, evolution and ecology are not going to be covered in this exam. So these I'm going to look at more in terms of the reasoning that we do, but know that the content for those isn't going to be stuff that you're held responsible for. So the first thing here, we want to circle one arrow that represents transcription. So transcription, if we remember, is going to be when we go from DNA to RNA. So I'm going to go ahead and circle that. So that step right there, going from the gene to the RNA, that would be a transcription step. And because I don't want to follow their directions, I'm going to circle 2 because I want to show you where both of those transcription steps would be. Hey. So either one of those would be acceptable to circle there. And I get a little bit paranoid whenever I'm doing an exam and I try to over-communicate. So it wouldn't be a bad idea to write in next to that this is where the transcription is taking place. So, oh, come on. Let's do, there we go. So just mark that, yep, transcription is taking place there, and remembering that transcription goes from DNA to RNA, and then translation goes from RNA to protein. Also, identify the molecule that would be absent if the enzyme YUCK is non-functional. So if TRPT is working, but YUCK isn't, this pathway is going to go until it gets to the thing that's broken. So tryptophan would get converted to I3PA. Enzyme YUCK is broken, so we aren't getting any IAA. So I'm going to quickly circle that as well. No, I'm not going to circle that. That's too confusing. Instead, we're going to say that IAA would not be present. So we'd get tryptophan converted to I3PA, but it would get stuck there. We need to predict how the deletion of one base pair at the 4th codon of the coding region would most likely affect the production of IAA. So we're looking at this enzyme right now, and we are looking at a deletion. So this is where we need to think back to translation, which was very recent. So the thing to remember is that codons are 3 base pairs long, and a deletion is going to alter the reading frame, which means that this is going to change every amino acid from the deletion onward. So if we lose stuff in the 4th codon there, that would radically alter this protein, and it would be very, very unlikely for it to be a functional protein. So deletion of the fourth codon will alter almost every amino acid and likely render the protein, the enzyme, non-functional. If TRPT is non-functional, tryptophan will not be converted to I3PA, and IAA will consequently not be produced. Whew, that was a mouthful there. So part C is asking us about a feedback mechanism by which a cell could prevent the production of too much IAA without limiting I3PA production. So this is where we would be looking at some kind of feedback inhibition setup. So this goes back to when we were talking about enzymes, reactions, and inhibitors, and all of that. So we would say something like the cell could use feedback inhibition in which IAA inhibits the activity of enzyme YUK. IAA could be a competitive or non-competitive inhibitor of enzyme YUK. I know that it's a little bit after I said we would be wrapping this up, but we'll go ahead and hit parts of D and E here, and then we'll call it a night. So part D, like I said before, this one being an ecology question is going to be beyond the scope of the exams, but we'll go ahead and look through this one anyway because it's still it's helpful to see the reasoning there. So identify the most likely ecological relationship between plants and rhizobacteria. This relationship where it's mutually beneficial is called mutualism. So that's the part that they wouldn't expect you to know. But we can describe an advantage to the bacteria for producing IAA. And that advantage is those plants release carbon-containing molecules into the root nodules. So the rhizobacteria are going to get carbon-containing molecules from the plant. And carbon-containing molecules, that's basically food. So what they've got going on here is essentially in exchange for nitrogen fixation and root growth stimulation, the bacteria are fed by the plants. So when you're looking at multiple things interacting, what you really want to do is think about how is one thing going to affect the other? Where's the benefit going to be? For that last question, we're looking at cheater bacteria. So cheater bacteria, they have the advantage that they're basically getting a free ride. The cheater bacteria gets food without having to expend energy producing IAA. So anytime you can save energy as an organism, that's more energy that it can then spend on reproduction or something else. So any place where they can cut corners and spend less energy is good for them. So these cheater bacteria have an advantage because they get a free ride here. We want to predict the change in the bacterial population that would cause the plant to reduce the number of carbon-containing molecules provided to the nodule. So basically, for this one, if the bacteria stop producing, or if too many bacteria stop producing IAA or fixing nitrogen, the plants, they would either stop or reduce the amount of carbon-containing molecules they send. So basically, with this one, you want to look at what is the plant getting out of it, and if it stops getting what it needs, it's going to pretty quickly stop giving what the bacteria need there. All right. Thanks, folks, for sticking with this, coming in here. If you've got particular problems or questions or things that you'd like to cover for the next one, go ahead and send me an email, leave a comment on Google Classroom, or find some other way to get that communicated to me. Next week, I think what we'll do is spend a little bit of time looking at some of the cell structure, so cell membranes, osmosis, diffusion, that sort of thing, and then do a deep dive into a problem or two from there. So that's kind of what we're looking at going through here. And let's see here. There we are. So yeah, thank you everybody for showing up here. And I'll see you guys next time. So stay healthy, be good to each other, and take care.